Okay, so today I'll be talking about my research, which focuses on the fungus-farming ants and their microbial symbionts, and how this symbiosis has driven evolutionary innovation in the fungus-farming ants. Okay, so this type of symbiosis that I study is an animal-microbe mutualistic symbiosis. And so what this means is that an animal and a microbe live in close association with each other, and both the animal host and the microbial symbiont mutually benefit from this interaction. And so we see these types of symbioses all across our planet. These are just a few examples. Many insects engage in these symbioses, including the fungus-farming ants, which I'll be talking about today. But we also see these within our own human bodies. Most people are familiar with the gut microbiome and its importance on human health. We also see these in sea animals, land animals, animals we farm for food. And so these symbioses are really widespread across our planet and important for animals, including us humans. And so despite the benefit that both of these symbiotic partners are receiving from this partnership, there is a challenge that arises from these symbioses. And the challenge is that if you think about it, an animal and a microbe are very different types of organisms, and they have very different selfish interests. And so somehow they must align these interests and maintain this symbiotic cooperation. And they must do this, maintain this cooperation and fidelity over long periods of time. So one of the ways in which they do this is through this evolutionary innovation of the evolution of what we call a symbiotic structure. And so what a symbiotic structure is, I'm just going to turn off the lights actually. What a symbiotic structure is a specialized structure on the host's body. So in the animal microbe symbiosis, it's on the animal's body. And this it serves as the interface where the symbionts can exchange chemicals. So you can think about it, the symbiotic structure is on the animal's body. This is typically where the microbe lives. And so this is where the animal and the microbe are in physical contact with each other. And so here they exchange chemical signals that allows them effectively to communicate and ensure that they'll receive their intended benefit. And so typically the animals secrete some nutrients for the microbes here, and then the microbe will secrete some sort of signal that allows the animal to detect that signal, and so that the animal can recognize the microbe as its symbiont and not just some random microbe or some pathogen. And so these symbiotic structures have evolved many times in a wide variety of animals. And so we see this repeated evolution, and that's what this figure is showing here actually from this review. It's showing the repeated evolution of these symbiotic structures in very different types of animals that engage in symbiosis with very different types of microbes. And so the repeated evolution of these symbiotic structures indicate their importance and their effectiveness at mediating these symbioses. And I just want to point out that these symbiotic structures, they have a vast diversity of morphologies. And so they can be a wide variety of shapes, sizes, even different locations on the animal's body. And so the thing that makes it a symbiotic structure is not necessarily its morphology, but its function of mediating the symbiosis. And so despite the repeated evolution that we've seen of these symbiotic structures and their importance in these symbioses, their evolutionary origins remain largely unknown. And so, yeah, their evolutionary origins. So how do these symbiotic structures evolve? What evolutionary mechanisms are involved in their evolution? Ultimately, and most importantly, what can we learn about the evolution of these symbioses in general from studying the evolution of these symbiotic structures? So these very broad questions are what I am attempting to shed light on with my research. And obviously, I can't study all these symbiotic structures, but what I focus on is the symbiotic structure of the fungus-farming ant and its evolutionary history. So the fungus farming ants are a part of a large complex symbiosis. And so I just want to point out right here that, the name fungus farming ant and the name attine ant are synonymous. And so I might refer to either one of these names, but I'm referring to the same group of ants. And so as the common name of this ant suggests, they farm a fungus. And so the ants will collect plant material and they'll bring back this plant material into their colony where they incorporate it into their fungus farm. And the fungus farm digests this plant material as nutrients, and then the ants will eat a part of the fungus farm. And so this fungus farm serves as the ants' primary food source. And so you can think of this fungus farm as like an ant's crop. And so similar to human crops, this ant crop is prone to pathogens. In this case, it's a specialized microfungus parasite called Ascovopsis. And Ascovopsis can infect the fungus farm, and a bad enough infection can, devastate the fungus farm, which can cause a collapse of the ant colony. And so the ants must protect against this pathogen in some way. And one of the ways that they do this is through engaging in an additional symbiosis with this mutualistic bacteria. It's an actinobacteria called Pseudonocardia. And so what the Pseudonocardia is doing is producing antibiotics that can inhibit the growth of this pathogen. And so it's a pretty significant service that the Pseudonocardia is providing to the ants. And so it's this interaction here between the ants and the Pseudonocardia bacteria that I focus on for my research. And so this is a mutualistic symbiosis. And as I mentioned, in a mutualistic symbiosis, both symbionts receive some benefit. So the ants are obviously receiving this really tremendous benefit from the bacteria in the form of antibiotic production. But what is the Pseudonocardia receiving in exchange? And so the Pseudonocardia lives on the exoskeleton of the ants. And if you zoom in into the exoskeleton, as you can see on this SEM here, what you'll notice is that their exoskeleton is completely covered in these symbiotic structures. And so these symbiotic structures are where the Pseudonocardia lives and where the ants provide nutrients for the Pseudonocardia. So we have this exchange of antibiotic production, and in return, the Pseudonocardia gets a nice place to live and nutrients. So it's a nice trade. And so these symbiotic structures they completely cover the body of the ant from the head all the way to the abdomen. And so what happens is that when the Pseudonocardia reaches its peak colonization, it completely covers the ant and actually turns it white. And so you can see the Pseudonocardia without a microscope because it grows filamentously, and you can see it as this white bloom. And so this is just to show that the symbiotic structures completely cover the body of the ant. So it's this symbiotic structure that I focus on for my research, and particularly try to answer this question of how did the attine ant symbiotic structure evolve, and what sort of evolutionary mechanisms were at play in the evolution of this structure. And to try to answer this question, I've been taking an Evo-Devo approach, Evo-Devo meaning evolutionary developmental biology. And so in short, the way the Evo-Devo approach works is by studying the development of your, in this case, my structure of interest, the symbiotic structure, you can uncover the developmental blueprint of your structure. So how your structure develops, as well as some of the key genes and proteins that are involved in the development of your structure. And then you can compare this developmental blueprint to other structures and other species to infer some sort of evolutionary history of your structure. So I set out to study the development of the attine ants, or the fungus farming ants. And in general, they go through a typical ant developmental cycle. They start as an embryo, then they go through the larval stage. They have several larval molts. After that, they have a pupal stage where they continue developing until they're finished developing and they close and they emerge as adults. and they go ahead and live their adult life. But really nothing was known about the development of the symbiotic structure, even when it develops. And so early on, I figured out that it's developing in this early to mid pupal stage. And so I point that out just to say that the developmental assays that I've been working to establish, as well as the images that I'll be showing in the following slides, mostly focus on this early to mid pupal stage. So one of the first techniques that I was able to get to work in the ants, it was this antibody staining technique. And so this really started to provide some insight into the symbiotic structures. And so what I'm showing here is an antibody stain of a pupa in that mid pupal stage, around 5 to 7 days old. And so this is the antibody stain. Here's the top of the head. Here's the eye antenna. And so it's like it's facing you. And so what I'm staining for in red is this HRP stain, which is widely used across insects to stain for neurons and the nervous system. In green is alpha-tubulin, stains tubulin, and in blue is hoax, which is similar to DAPI in that it stains the nuclei. And so what you might notice here is that the head is just loaded with these red dots. And so this was really exciting to me because I recognized these red dots. I recognized the distribution of these red dots. And to me right away, I could identify these dots as a similar distribution to the symbiotic structure. So to show you the distribution of the symbiotic structure, I'm showing this SEM. This is actually of an adult ant, but I'm just showing an adult because you can clearly see the final distribution of the structure. And so you'll notice these lines, those are the sensory hairs. But if you look even closer, you'll see that the head is just completely filled with these little dots. And so each single dot corresponds to an individual symbiotic structure. And so, as I mentioned before, these symbiotic structures completely cover the body, and this includes the head, where it just completely covers the head. And so the abundance of the symbiotic structure from this SEM and the abundance of these red dots are very similar. And so these red dots being the symbiotic structure becomes more obvious when you zoom in. And here you can see each individual symbiotic structure. And you can also see the connecting neuron of each individual symbiotic structure. And so once again, in red is this HRP stain that is staining for these neuronal cells. And so what you'll also notice are these long green lines. These are sensory hairs. The ant head is also sort of sparsely populated with these sensory hairs. And this was a nice confirmation because the sensory hair is a type of sensory organ, and it's very well documented that these sensory organs are also innervated. They also have this neuronal cell. And so you can see the neuronal cells of each sensory hair as well. And so this was good confirmation that the HRP is working as intended in the attine ants. And so this is a more 3D view. And once again, you can see each individual symbiotic structure. You can see the neuronal cell of the structure. And this kind of gives a perspective where you can also see what I assume would be the dendrite extending apically and sort of penetrating the surface of the cuticle. And so what was really interesting to me is the fact that both, and I should point out, this is also the hair that I just talked about, this green line. And so what was really interesting to me is that the symbiotic structure and the sensory hair have this similarity in that they're both innervated cuticular structures, meaning that they're innervated, they have this connecting neuronal cell, as well as they're cuticular, meaning they're located on the surface of the cuticle. And so I propose this hypothesis that the attine ant symbiotic structure is a co-opted / a modified sensory organ. And so another way to say this is that the sensory organ and the attine ant symbiotic structure are homologous structures. They have this shared homology. And so to approach this question of homology, I'm using what I had mentioned before, this Evo-Devo approach. So with the Evo-Devo approach, I would compare the developmental blueprints of the sensory organ to that of the attine ant symbiotic structure. And so by developmental blueprint, I mean the step-by-step cellular divisions, including the final differentiation of each component of the structure, as well as the key genes and proteins that are involved in each of these steps. So if this hypothesis is true, that the symbiotic structure is a modified sensory organ or sensilla, I would expect that the developmental blueprint of the ant symbiotic structure would closely resemble that of the sensory organ. And so you can think of this co-option mechanism as sort of taking an existing developmental blueprint and sort of copying and pasting it into a new developmental context, whether this be a new time or location in development. And obviously there would be some differences, because in the end you end up with structures that are different from each other in function and morphology. However, what would be expected is that a large part of the blueprint would remain the same. So you would see this resemblance. And this is evident in the evolution of sensory organs. Sensory organs, they have been really important, not only in ant evolution, but in insect evolution as well. And they are used by insects to sense external stimuli in the environment. And so if you can think of some external stimulus, the ants probably have some sensory organ that can detect it. So as I already mentioned, the sensory hair is an example of a sensory organ. It detects mechanical touch, but there's also sense organs that can detect odor, humidity, temperature, stability during flight, and on and on. And so what I learned from the literature is that even though these sensory organs have diversified greatly, they generally still follow this core developmental blueprint. And so fortunately, the developmental blueprint of the sensory organ is very well characterized in the literature. And so what I've been doing is trying to characterize the development of the symbiotic structure so that I can compare and see if there is this resemblance. And so far, I've seen a number of similarities in the development of the ant symbiotic structure and the sensory organ. And so the first developmental similarity between these two structures is what I had already mentioned, the innervation. And this includes the differentiation of this neuronal cell, as well as sort of connecting to a more central nervous system, as you can see up here. If you look at one of these symbiotic structures, you'll see it connects to a more central nervous system, same with the sensory hairs. So this is great. And so what I try to do next is try to characterize some of the gene and protein expression during development. And I just want to mention that in the following slides, I'll be sort of using this sensory hair, which again is a type of sensory organ. I'll be using this sensory hair to compare to the symbiotic structure. And the reason for comparing the symbiotic structure to the sensory hair is because, like I said, it's a type of sensory organ. It follows this developmental blueprint that's characteristic of the sensory organ, but also it's just very easy to identify in these stains, and they have very large nuclei, so it makes the imaging very easy. Okay, so to characterize the gene expression, I've been using this assay called hybridization chain reaction, or HCR. And so this HCR method uses fluorescent probes to visualize the expression of your genes of interest. And so here I'm showing an image of HCR. This is a region of the pupil head. It's zoomed in. And so in orange, I'm showing the expression of this gene cut. And in blue, I'm showing this dye hoaxed, which again stains the nuclei. And cut is a gene that's well known in the development of the sensory organs. And so what you might notice from this HCR image is two things. One is that there's these regions these smaller regions of cut expression, as well as these larger regions of cut expression. And so if we look again at an SEM This is of the exoskeleton of an adult, just to show the distribution. But we see that these smaller regions pretty closely resemble the abundance of these smaller regions of cut expression, pretty closely resemble the abundance of the symbiotic structure on the head of the ant. And the abundance of these larger regions of cut expression pretty closely resemble the abundance of the sensory hairs on the ant's head. And so the reason that the sensory hairs have these larger areas of expression, here is a stain showing wheat germ agglutinin, which is stains the sensory hair, and then hoax to stain the nuclei. And we can see that these sensory hairs just have giant nuclei compared to the surrounding nuclei. And so that's why they have these, very large areas of cut expression. So this is the only gene that I've characterized so far with this method. I just started to get this working in the ants, and so that's sort of the trials and tribulations of working on non-model organisms is trying a bunch of different methods, and a lot of them won't work. But this HCR seems to be very promising, and I just got it to work a few weeks ago. So in the future, I'm very excited to try this method with a lot more genes. Okay, so moving on to protein expression. To characterize a protein expression, I'm using, once again, antibody staining. And so just to describe what's going on here, on the left is the attine ant sensory organ. Once again, I'm using the sensory hair to sort of represent this typical sensory organ development. And on the right is the ant's symbiotic structure. And so in green, I'm showing the expression of the protein cut, which I just talked about with the HCR data, same gene, but this time I'm showing the protein's expression. And then the other colors are the same colors I've been using. White is alpha tubulin, red is HRP, blue is hoax, which again stains the nuclei. And so, I mean, it's pretty obvious that the cut expression in the sensory organ very closely resembles that. Very closely resembles the cut expression in the ant's symbiotic structure. And so in sensory organ development, in the sensory organ, this cut expression is being expressed in the two most apical cells. So the cells closest to the surface of the cuticle. In a sensory organ, this is called the socket cell and the shaft cell. The shaft becomes this hair shaft and pushes through this socket cell. The socket cell forms this invagination and the shaft pushes through it. And so we see a similar cut expression in the symbiotic structure in these two most apical cells. And so a second gene that I've characterized protein expression is this gene called the Keat-Skute homolog. And so on the left here is, again, the sensory hair representing sensory organ development and the ant's symbiotic structure. And again, we see very similar protein expression of this gene. This gene is very well known to play an important role in sensory organ development. What it does is it marks the progenitor cell. The expression of this gene marks the progenitor cell, and this progenitor cell or mother cell divides and becomes the entire structure. And so essentially, if you knock down this gene, as has been done in other insects, you will get a complete absence of these sensory organs. And so it plays a very important role, and it's quite interesting that we see a similar expression in the symbiotic structure. So the third and final developmental similarity between the sensory organ and the symbiotic structure is just the general developmental sequence. And so what I'm showing here on top, obviously this is a diagram, but this is the typical sensory organ developmental sequence. And then on the bottom, I'm comparing some of these steps to what I've seen so far in the symbiotic structures development. And so I'll go through this step by step just to explain the colors that I'm using. In green is, again, this Keat-Skute homolog antibody. Red is HRP, so this is staining for neuronal cells. Blue is staining the nuclei, and white is alpha tubulin. And so this first step that we see here, this is a 2-cell stage. this is not too exciting in itself, but the reason I'm showing this is because just to show that this acute-skewed homologue is present very early in development. And so, you know, it serves as a nice marker when I look at an early stage pupa to see where each of these symbiotic structures are developing. The HRP stain, which I use to stain the neurons, is also a good marker, but it doesn't develop, it doesn't differentiate until this later stage. So this acute-skewed homologue serves as a nice marker early on in development. And so moving on, the 3-cell stage, not too exciting again in itself, but this is just to show a similar pattern where these 2 cells here was the basal cell, and this divided. And so at the 3-cell stage, the next cell to divide is this apical cell that's similar to what we see in sensory organ. But it starts to get interesting in the 4-cell stage, and in the symbiotic structure, in the sensory organ, what happens is there's the formation of that socket cell that I had mentioned early on in the hair. This is the invagination that the hair shaft will push up from underneath. And I think that I'm seeing a socket cell formation as well in the symbiotic structure. This is sort of more indirect evidence because what I'm seeing here is a lack of acute-skewed homologue protein, but I see it pretty consistently across the symbiotic structures. There are better ways to visualize this, which I've been working on, but this is sort of indirect evidence for now. And then this last step here is the differentiation of the neuronal cell. At this stage in the sensory organ, we see the differentiation of this neuronal cell, and then the dendrite extends apically and pushes up through the structure and eventually penetrates the cuticular surface. And so this is what we also see in the symbiotic structure. This is a top-down view, so you're looking down on the structure. And so this neuronal cell is the most basal cell, and you can see it pushing upwards through the structure and eventually penetrating the surface. So that is all that I have today. And so to sort of summarize, I've studied, some aspects of the development of the ant's symbiotic structure, as well as the sensory organ, and I've seen some developmental similarities in these 2 structures. First of all, they're both innervated cuticular structures. They share some important developmental genes and protein expression during the development of these 2 structures. And their overall developmental sequence is pretty similar as well. And so I think that with the evidence I have, it is pointing towards this hypothesis that the symbiotic structure is perhaps a co-opted or a modified sensory organ. But there's obviously a lot of work that I need to do still. And so what I have planned for future directions is to characterize the expression of more genes of interest during symbiotic structure development. As I've mentioned before, the sensory organs development is very well characterized. So there's a long list of genes that are known to play a role in the sensory organ development. And so using this HCR method that I've just started to get working, I will be testing the expression of these genes of interest in the symbiotic structure. Secondly, some sort of gene knockdown method, RNAi or something like that, would be nice to validate the function of these genes in the development of the symbiotic structure. This is something that I've been working on, but this is not totally optimized yet. And then lastly, as sort of a non-genetic final approach or future approach, I would like to do some sort of functional analysis of the innervated symbiotic structure, something like single sensillum recording. This is what I'm showing here in this image. And so because this is done all the time in the sensory organs, so because the sensory organs are innervated, you can stick an electrode in a single sensory organ, and you can expose it to some stimulus, to an odor or whatever stimulus you choose, and you measure the electrical activity of that sensory organ. And if indeed the stimulus you expose it to is what the sensory organ is responsible for detecting, you'll see an increased peak of electrical activity compared to some negative control. So it would be very cool to do this in the symbiotic structure to see if the symbiotic structure responds to some chemical that the Pseudocardia bacteria, its symbiont, is producing. So yeah, so just some acknowledgement. The Curry lab, my PI, Dr. Cameron Curry, the Sharma lab, Dr. Prashant Sharma, and especially Emily Seton and Guy Gannett, his two grad students that have really helped me establish these developmental methods in the ants. None of these methods were really established in the ants, and so I had to figure out these methods by myself, but definitely wouldn't have been able to do it without their help and guidance. And my committee, who's provided great guidance so far, and also Sarah Swanson and Bill Schneider for help with some of the imaging. So thank you to everybody for being here and for listening, and I could take any questions that people might have. Yes, Guy.
Amazing talk.
Thank you.
Very nice to see. So I have a few questions, but I'll just ask 2 questions now. Do you know what time the bacteria colonizes the symbiotic structure?
Yeah, that's a good good question. I think I had a backup slide here. I don't know if I'll be able to show it, but they actually colonize the structure in the adult stage. So the symbiotic structure forms here in early to mid pupal stage, and then the Pseudocardia colonize the ant. And there's a very specific window. It's like right when it closes until 2 hours, the Pseudocardia have to colonize the ant. And if they don't, the ant won't be colonized in the future at all.
Okay. Yeah, I guess that answers my second question.
Okay.
I was just wondering if if you have like a marker that you could stain the bacteria and the gene expression at the same time, because I was wondering if during the development of the symbiotic structure, the bacteria could be influencing the expression of the genes, but I guess that kills my question, right?
Well,
it cannot be there before the structure forms.
Right. But I also still wondered that, like, if a worker ant that is taking care of the pupa, if perhaps the bacteria on the worker ant releases and is picked up by the pupa. But in the Curry lab, we have an ant colony that somehow, by being in lab, it's lost its Pseudocardia bacteria. So when we collected it, it had the bacteria, and now it doesn't have the bacteria. So I've also tested the development of the symbiotic structure in that colony. So that way I know that even the ants taking care of the pupa don't have the bacteria. And so even in those pupa, the symbiotic structure develops, from what I can tell, perfectly normal. It's innervated.
There's no bacteria
in the whole colony, yeah.
The symbiotic structure develops normally.
Yeah, from what I can tell.
Cool, right?
Yeah.
Yeah, cool. I'll just...
Okay. Emily?
So you keep saying we have this big literature, this big list of candidates, and all these things we can check. Well, now that you have HCR, which gene are you most excited about finding an expression pattern, and why?
So I'm really excited about this gene called this Akitsky homolog that I showed the protein expression for. But I'm really excited for this gene because in other papers that I've seen done in insects looking into the evolution of the sensory organ, this gene seems to be a very important gene because essentially if you knock down this gene, the symbiotic structure the sensory organ won't even form. So it's very important in that first cell. And so if I can see some expression of the gene in the first progenitor cell of the symbiotic structure, that would be really exciting, especially if I get the gene knockdown method to work, to sort of validate with that. And then basically, you know, the dream, I guess, would be to knock down this acute-skewed homolog in the ants and essentially get an ant that has no symbiotic structures formed at all. Kirsten?
Would you think the ants are something from bacteria? And do you think it has any role in why there's less effect on ants?
Okay, so what do I think the ants are sensing from the bacteria? I don't know exactly what it would be, but I think that it would be just some chemical that the Sunicardia produces. Either it could be sort of direct or indirectly, some indirect chemical or some direct chemical. And I think that as I was talking about to Guy, I think the function of detecting this chemical could be, sorry, this is going slow, could be this short 2-hour window that the Sunicardia must colonize the ants. And so if the ants detect some chemical from the Sunicardia and they know it's on the exoskeleton, they'll continue to release the nutrients that they release for the Sunicardia. And if they don't detect the Sunicardia in their symbiotic structure from some chemical that they're releasing, then perhaps they shut off the nutrients in the symbiotic structure, which would make sense because you don't just want free-flowing nutrients on your exoskeleton that perhaps pathogens could sort of take advantage of. And sorry, could you say your second question again?
Oh yeah, so like how would you think the structure changes if there's less bacteria on it?
Oh, right, okay, so.
Sorry, I kind of reworded it.
No, that makes sense. So yeah, so as Kirsten mentioned, this is what the ants look like when the Sunicardia reaches peak colonization. But as the ants start to age, the bacteria sort of reduces to the ant's propleural plate, which you can sort of think of as like the chest area. And so if you look at an old ant, the bacteria is only present on the propleural plate. And so what I would like to do is to antibody stain in old ants. So I would somehow have to section the ants and antibody stain it. And it would be interesting to see if the symbiotic structures on the head and the abdomen remain innervated, even though there's no bacteria there. I would guess that the neuronal cell perhaps would maybe die off, just because I would think that's sort of expensive to maintain a neuron. And so I would expect that the neurons would still be there in the symbiotic structure on the propleural plate, but perhaps nowhere else on the body of the ant. Nicole.
So, are there related ants that don't form this symbiotic relationship with this? What do their people look like? I mean,
Yeah.
You know, I guess what I'm trying to ask is, are the symbiotic structures coming at the expense of the sensory organ? You know, so would the other ants have more sensory organs?
Yeah.
Do they have any structure that resembles this?
So there are ants that are fungus-farming ants that have lost the Sunicardia or don't form association with the Sunicardia. I'm forgetting how many species of fungus-farming ants there are, like over 200, and a lot of them don't actually engage in the symbiosis, but I would say most of them do. And so the ones that don't engage in the symbiosis, they just take extra care of the colony. They're very clean. But the ones that don't have the Sunicardia, their exoskeleton just looks very smooth, and there's no symbiotic structures there. So essentially, when you lose the Sunicardia, you also lose the symbiotic structures. What's interesting is like kind of what you're asking. Does it come
phylogenetically in the opposite direction? Instead of going, sort of deeper into these ants, what's the step back a branch?
Oh, okay. So
yeah.
Closely related to the fungus-farming ants. I mean, there's
prior to the evolution of the symbiotic structure.
Right. I mean, yeah, nothing. From what I can tell, there's no like compensation in additional sensory organs in sort of the sister group to the fungus farming ants. What I have noticed is that a lot of time ant heads they have a lot of hairs that cover the head, if we're just looking at the head. And in the ants that have the symbiotic structures, there does seem to be perhaps some correlation, like the heads are less hairy, you could say. So perhaps at the expense of having all these symbiotic structures, you do lose some hairs or some other sensory organs across the body. But I can't say that like for sure. This is just sort of a hypothesis.
I'm just wondering about this sort of patterning, if your hypothesis is right, and co-opted a set of genes that are actively involved with the sensory organ, what decides which cells are then going to become sensory organs? Which ones are going to symbiotic structures? Right.
I mean, yeah, I would love to work out these details eventually. But it could just be that the developmental plan is sort of co-opted into a new developmental timing or location. So locational cues signals that these will be symbiotic structures, these will be sensory hairs. But right now, yeah, I'm not sure. But I'd love to work out those details eventually. Yes.
So you mentioned that older ants, they have a bacteria growing in their chest plate.
Mm-hmm.
So it got me wondering, does the colonization of bacteria start at any particular part of the body, or does it just grow uniformly all over? Does it like start to decolonize in a particular area as well?
Yeah, those are good questions, but I honestly don't know. I don't know if the dynamics of those are worked out at all. But it is interesting to know perhaps where it starts colonization and sort of how it retreats. But yeah, I'm not sure. Siddharth?
Are there any closely related ant species where they have the symbiotic structures but don't have fungal colonies?
Don't have the symbiotic bacteria?
Yes.
There are a few examples of that. It's one single clade where they've lost the symbiotic bacteria, but they still have the symbiotic structures. I wish I had a picture, but the symbiotic structures do seem very reduced. Like it's even questionable if you can call them a symbiotic structure, but it would be interesting to look at those species to see if they are still innervated and sort of if they still have all the components of the symbiotic structure. Okay, thank you, everybody.